Hi, welcome back. Before the break, we were looking at the work that Gregor Mendel did in terms of finding out how plants inherit their characteristic. Now, you must remember that although he worked with pea plants and the original work was all done with plants, all of this, of course, applies to you as well. And all of those characteristics that we spoke about earlier, like your ability to roll your tongue, or whether you have a widow's peak or a straight hairline, all of these things can be worked out in exactly the same way. And as you study genetics, you're going to come across lots of different problems in past exam papers or in your textbook that your teacher's going to give you to solve. But all of these problems, irrespective of what factors you are looking at, can all be solved in a very systematic process, which we're going to look at very briefly now. These are what we call monohybrid crosses, where we take one characteristic that has the two competing alleles and we cross them and see what the result's going to be. And you may have seen that your teacher asks you to draw what is called a Punnett square. It has a capital P because it's named after the person who developed this very systematic and easy way to work out how genetics crosses work. Now, when you do a Punnett square, it's like a maths problem. You know that you've been told always show all your working out in a maths problem, so that if you get to the wrong answer at the end, maybe you can still get some marks for the way you worked it out. Exactly the same thing applies here with genetics problems. Start off always showing what the parental generation is. Always write down the phenotype, which is what it looks like, followed by the genotype, which is what its genetics are, what its genes are made up of. Always write it out with the cross sign in between so that people can see you are crossing a double tall with a double dwarf. Then you have to show the segregation of the alleles into gametes. So you take this parent and you say that the possible combination of alleles in the gamete can be capital T, capital T. In this parent, the possible combination of alleles in the gametes, small t, and you work the cross. Capital T, small t, that is what will be the possible combination in the new offspring. And you work that out for each possibility, capital T, small t, capital T, small t, capital T, small t, and then you show the first filial generation, which is also called the F1 generation. And we see, first of all, the genotype. We say that there is 100% capital T, small t. In other words, 100% heterozygous offspring. And remember, because of the law of dominance, a heterozygous condition will always show the phenotype of the dominant characteristic. Now you have to take that into the second parental generation, where you're going to do a self-cross. You're going to take those parental, your new parentals are going to be the old first filials, and you're going to draw your Punnett square again. You're going to take the one parent and segregate the alleles. You're going to take the other parent and segregate the alleles, and then you're going to do the cross. And this time you get a very different result. Capital T, capital T. If we did this cross here, capital T, small t, this cross is going to result in capital T, small t, and small t, small t. So our second filial generation, the results of this cross, 1 capital T, to 2 capital T, small t, to 1 small t. That is our genotype. What does that translate to in the phenotype? We say that those individuals will all be tall due to the law of dominance, and this individual will be dwarf. which can translate into 3 tall to 1 dwarf, or 75% tall, 25% dwarf. And as I said earlier, remember that you will get a mark for each of these different steps that you show in your working out, not just for the answer at the end. Students often get very panicky and they say, oh, there's so many options that there possibly could be. In fact, there aren't. There are 6 possible options. You can either start off with the parental generation as all dominant, which will lead to an all dominant genotype and an all dominant phenotype. You can have the opposite of that, where you start off with a parental generation of all recessive, which means that you're going to get an all recessive offspring. You can have one dominant homozygous crossed with a heterozygous, and you're going to get all dominant phenotype, because remember that clearly is dominant and so is that. You're going to have another option of having a capital A times small a. They will all be the dominant phenotype again. You could have a heterozygous crossed with a heterozygous. That's just the cross that we showed you now. And that's when you get your 3-to-1 ratio in the phenotype. And if you have a heterozygous crossed with a homozygous recessive, you're going to get a 50-50 or a 1-to-1 expression of those genes. And you need to play with lots of different examples. You need to work through lots of possible problems with your teacher through the textbook. The textbook should have lots of examples for you to pursue so that you can get practice at working out these problems for yourself. You'll find, just like with maths, if you drill yourself with the problems, they will become easier and easier. Read what the question is and use a Punnett square to help you segregate your alleles, do your cross, and follow that systematic procedure. I hope that you've enjoyed our exploration of inheritance and genetics. Next week we will be looking further into genetics, so I hope to see you then. Goodbye.